Continuing with chapter 5 on membrane dynamics. In this chapter, we will be talking about all sorts of transport processes, including osmosis and tonicity, diffusion, protein-mediated transport, vesicular transport, epithelial transport, and the resting membrane potential integrated membrane processes that we'll leave for later because that's important as our transition to unit 2, which will be on the nervous system. But so we'll start out here with sort of a reminder about the 2 fluid compartments that we just talked about in chapter 3. So we have the extracellular and intracellular fluid compartments, and the cells are separated, or the separation line between the extra and intracellular fluid compartments is a plasma membrane. And so we will be talking here about osmotic equilibrium, chemical disequilibrium, and electrical disequilibrium. So equilibrium means the same on both sides. In physiology, a lot of times we don't want that. And homeostasis a lot of times means that we are able to separate, let's say, ions from one another. So we need to have a strict separation of sodium and potassium between the extra and intracellular fluid compartments, and much more to come on that end. So, but here, the fluid compartment, sort of this overview. Whatever is inside of the cells, the fluid inside of cells, that we call that intracellular fluid compartment, the ICF. The extracellular fluid compartment, that's all the fluids between the cells, we call those interstitial fluids. And then the blood plasma, of course, contains also some fluids that's outside of cells. Here, this figure, you can see sort of the distribution of body fluids on an average person of 70 kilograms. The intracellular fluid compartment is about 2/3 of total body water, so the ICF is much bigger. And the extracellular fluid compartment contains about 1/3 of your total body water. We have a little bit of water in the, relatively speaking, in the plasma, and then in the interstitial fluid, you have about 75% of your extracellular fluid is between cells. I know I've shown you this before, but to be in homeostasis, you need to be able to separate these ions. The most important ones would be sodium and potassium. So we don't want an equilibrium. We don't want equal on both sides, okay? So we want a disequilibrium. And so therefore, this membrane needs to function to enable us to separate these ions and sort them out and establish very strong concentration gradients. In the intracellular fluid, you want to have a lot of potassium, so the purple one is very important, and very little sodium. You see that tan one, or light orange, that one is sodium. You want very little inside of cells, but you want most of the potassium inside of the cells. In the interstitial fluid and in the plasma, so the extracellular fluid compartment, it's exactly the opposite. You want a lot of sodium and very little potassium. You might think, no big deal, I'm just going to add a little bit extra potassium. Nope, it is a big deal. It's a huge deal. If this potassium concentration rises above 5 millimoles, you are ending up with cardioarrhythmias. If it gets a lot more than 5 millimoles, then you're dead. It's not okay. So the potassium concentration has to be very carefully monitored in the extracellular fluid compartment, and we talk a lot more about that when we talk about action potentials. So let's first start on this transport lectures. Let's start with osmosis. That's very important. So the body is mostly water, and we are literally water-based people. And the thing is, though, that water will go down its concentration gradient. So if you have a different concentration of dissolved particles between 2 compartments, and water is able to cross a membrane or go between these compartments, then water will go down a concentration gradient, and that's known as osmosis. So Osmosis is defined as the movement of water across a membrane in response to a concentration gradient of dissolved particles. And the movement of water through these biological membranes is through aquaporins or simply by diffusion. A little bit can diffuse. Okay. Osmotic pressure is basically how steep is the concentration gradient. In other words, how much osmotic pull do you have? If there's a very high salt concentration, let's say in one compartment and not much on the other, then you're going to have a huge osmotic pressure. So it's the pressure of water wanting to go down its concentration gradient. We're not going to pay too much attention to define molarity versus osmolarity. You can pretty much interchangeably use that. It's if you do IV solutions and you calculate how much of certain substances you need, and you need to calculate osmolarity because it's how many particles you end up with. For example, sodium chloride dissociates into sodium and chloride. There you have 2 particles, versus molarity only expresses the concentration of salt that there is and not how many items they will dissociate into. Okay, anyway, so osmosis is the movement of water down a concentration gradient. And we need to clarify a few more terms here along those lines. Isoosmotic, hyperosmotic, or hypoosmotic. Again, I tend to use these terms tonicity and osmolarity interchangeably. So I usually talk more about isotonicity, hyper and hypotonicity. So I use more of these terms right here. The tonicity is this. Now let's take a look at how all of this works. So here first, a little comparison. How much water is in the human body? So you're best hydrated when you are an infant, and then I always say we go from grape to raisin. Basically, we're sort of slowly drying out, and by the time you're 60 or older, your body weight by the percent of total body weight that's water has dropped significantly. So you're starting out with 65%, and then you're down to, on females, even more, 46%. So a significant raisin state here, pretty dehydrated. Anyway, that's just a joke. So let's review this here one more time. The different fluid compartments. Again, I think we had that earlier, same thing. So when we talk about osmosis, we're going to consider how water moves down a concentration gradient, and in the human body, how water moves between these fluid compartments. And so this is the typical representation of intra and extracellular fluid compartments. And here again, same thing. About 2/3 of your total body water are inside of cells. That's the intracellular fluid compartment right here. And then the extracellular fluid compartments, the remaining 1/3. And most of it is in the interstitial spaces between cells, but some of it is also in your blood, forming the plasma. Anyway, so here we go, osmosis. Now, osmosis, think of it as just water going down a concentration gradient. You will never make a mistake on an osmosis question if you think of it as water going down a concentration gradient. So that means you're looking at the water concentration as opposed to the dissolved particle concentration. But anyway, so the idea here is that if we have 2 compartments, A and B, and there's a higher particle concentration in compartment B, then naturally there's less of a water concentration in compartment B. So here the water concentration in A is higher, the particle concentration is lower, and in B, the water concentration is lower and the particle concentration is higher. Now, as I said, you'll never make a mistake if you consider the water concentration. Now, clearly the water concentration is higher in A. So what's going to happen is that A is going to lose water, and it's going to go to B. So we're trying to dilute B, the compartment B, to the point where we have an equilibrium, where we have the same particle concentration and water concentration, that is, to the point where there is no more net flux. There's no more concentration gradient, therefore no more osmosis. And of course you can represent it in different ways. Here in this case, they expanded basically B. There's water going from A to B, and it expands the whole compartment B to the point where we then then have equal concentration and no more osmosis, no more net movement going. Okay, so let's consider this table right here. So we have different solutions, and so if we have solution A, which is 1 osmole of glucose, and solution B is 2 osmoles of glucose, and solution C is 1 osmole of sodium chloride. Now, how to figure out which one is hypo, and iso osmotic to each other? So we can say that C and A, C is 1 osmole of sodium chloride and 1 osmole of glucose. We just consider sodium chloride here as a 1 particle. Then we have to say that C and A are isoosmotic to one another. Now, B is 2 osmoles of glucose, so it is hyperosmotic to A. It's also hyperosmotic to C. Anyway, I think you get the idea of this. I'd like to bring in other examples as well, so we'll just go through these book examples and then I'll talk to you more about this. Now, let's consider what happens to cells. When we place a cell in distilled water, so then outside of the cell, if the cell is placed in distilled water itself, of course, contains particles. And so that means if we're placing the cell in distilled water, then the water concentration is higher outside as opposed to inside. And that means water will go down its concentration gradient from the area of high, 100%, outside of the cell down into the cell, where it's somewhat less than 100%. So that means the cell will swell. Water is going to rush in, and if we don't have cell walls to protect our cell, then the cell will even burst, and I'll show you that. So if the cell swells, then we know that the cell was in a solution that was hypotonic. And if the cell doesn't change size, that means the cell is sitting in a solution that's isotonic, same ionic strength, no concentration gradient. And if the cell shrinks, then we know the cell must have been in a hypertonic solution, because now the water concentration is higher inside of the cell, and so the water is leaving the cell to dilute the outside. It has a higher concentration of dissolved particles. Okay. So the following slides here, they are going into a little bit more detail on the tonicity versus osmolarity kind of issue. And it is a big deal when you're making IV solutions. And you can there was another slide right here. You can take a look at these slides. I'm not going to ask you specifics about the difference between tonicity and osmolarity. So I just kind of want to move on here. You can take a look at these rules for osmolarity and tonicity and how that makes a difference when you're making IV solutions, where you need to pay attention to how many dissolved particles you have in your solution, because it counts for which way the water is going to move. So I want to move on here to transport processes. And so we're going to take a look at different diffusion mechanisms. We will be considering bulk flow, which goes strictly down concentration gradients, which basically doesn't have any kind of barriers. So where there we just have concentration gradients, and these gases such as CO2 and oxygen, they will go down concentration gradients without barriers. And it's also sometimes true for liquids. For the most part, though, we need to cross membranes in the human body, and so there we need to give the cell the ability to be selective, so it can decide what it wants to let in or out, for that matter. And so we have this selectively permeable membrane. It does allow for bulk flow of gases such as oxygen and CO2, but other than that, the cell is pretty picky and wants to decide what goes in and what goes out. And so the all-important term here for these transport processes is concentration gradients. Okay, well, do we have concentration gradients? Yes, and the answer is very clear on this. In the human body, there are many concentration gradients. Concentration gradients allow for diffusion mechanisms to happen And the great thing about diffusion mechanism is that it doesn't cost you any energies. All you have to have is a concentration gradient, and then you can do all of this. You can do all of this passive flow. All you have to have then is sort of a window or a door, like a pore in your cell membrane, that allows the stuff to go down its concentration gradient, and you get great movement of stuff for free. You don't have to put in any energy, and that is really the amazing thing. In the lab, I have this diagram that I always do. You should reference that as well. So this lecture here goes along very well with the lab that you're supposed to be doing for this week. Active transport processes, they always involve ATP, but then the good news is you don't have to have a concentration gradient. So these would be exo and endocytosis, and all of the ion pumps, they are all driven by ATP, but then you move your stuff and you don't have to have a concentration gradient. Now, passive processes, those are the ones that your cells and your body absolutely love, because all you have to have is a concentration gradient, and then you move your stuff for free, energetically for free, that is. Okay. So diffusion properties, it is a passive process, and therefore it does not require any ATP input. The only thing that you must have is a concentration gradient. So we're always allowing the flux of a substance from the area of high to the area of lower concentration. And the flux or the diffusion will stop once you have an equilibrium. If there's the same amount of dissolved particles on both sides, osmosis, for example, will stop. Okay, so once you have an equilibrium, no more diffusion happens. Diffusion is rapid over short distances. It is directly related to temperature, and in the lab you will see some experiments. Well, you can also picture that. Think about it. If you put a sugar cube in your iced tea versus your hot tea, where even if you don't stir, in which glass is the sugar going to dissolve faster? And it's clear. The higher temperature will cause diffusion to happen much faster, because diffusion is directly related to temperature. There's a direct relationship. Diffusion is also inversely related to molecular weight and size. So that means if a very bulky, big molecule is going to diffuse at a lower rate as opposed to a small molecule. And in open systems or across a partition like a cell membrane, diffusion is great. It will move stuff without the input of energy. Again, as long as you have that concentration gradient. That's the must-have thing. You must have a concentration gradient. Okay. This is an experiment that you would have done in a real lab, but since we're remote here, you're going to just get to see that. It's Petri plates. They're filled with agar, and you are drilling 2 holes in them, and then you're filling the 2 wells there with 2 different dyes, and then watch them diffuse into the agar medium. What happens is that a small molecule, such as potassium iodide, is going to diffuse much faster. You can see there's a bigger circle surrounding the well, much faster than something as big and bulky as Congo red. In the lab, we are actually using the methylene blue as the bigger molecule, and we have the potassium permanganate as the smaller one. But you can see how molecular size and weight makes a difference here. In terms of rate of diffusion, the big bulky Congo red is going to diffuse much smaller, much slower into the agar. Okay, so here's some rules for diffusion that you can take a look at if you like. And so let's move on here. Lipophilic molecules, something that dissolves freely in the phospholipid bilayer, they will be able to cross cell membranes freely. So they don't have any barrier. So anything that is has no polarity, is small or lipid in nature, will cross the phospholipid bilayer with no barrier. And here are some examples that are shown. So here's your typical phospholipid bilayer representation, and it shows you that the very small molecules, they can cross. Anything that's lipid soluble will be able to cross without problem. But anything that's larger or charged or that is hydrophilic is not going to be able to cross the phospholipid bilayer, and you don't need to know these diffusion laws. That's not necessary. So the function of the plasma membrane, we already talked about that earlier. That is, we have this phospholipid bilayer, and there are many proteins embedded in that phospholipid bilayer. And there are also enzymes embedded in there, and very important for physiology, these membrane receptor proteins. And we have transporters such as channel proteins and carrier proteins. All of them are very important for physiology. So here's a chart that shows you all of those different types of membrane proteins, and for physiology, these transport proteins are very important. So I highlighted here membrane transport, carriers, channels, also the gap junction channels that were important. And then we have different types of ion channels. They have different gating mechanisms. Very important for physiology are the voltage gated channels, and then also chemically gated channels. We only have one example of a mechanically gated channel, such as over here. But anyway, so let me show you some of these membrane proteins. And so here's a representation of a receptor membrane protein. And so here would be a ligand that's binding here to this receptor, and then once this interaction has occurred, then events in the cell will happen. We'll talk much more about that. There are also some of these receptor ligand complexes. They will actually be internalized inside of the cell. We'll talk more about that. Now let's first here go back to channels. We have water channels, we have ion channels. There are open channels, and then there are gated channels. And again, very important for physiology are voltage gated ion channels, and then we have these chemically gated channels that will respond to, let's say, a neurotransmitter. We also have sometimes mechanically gated channels. Okay, so here are some representations of how these transport channels might work. Here is a channel that's just open right there at this point, but you need to have the ability to control what goes inside and outside of cells, because if we just let things go down a concentration gradient, well, then we can't really take advantage of the concentration gradients anymore. So we need to have the ability to allow flux only when we want it as a cell. And there are different ways you can do that. A popular way for cells, for channel proteins, is to have a little gating mechanism that closes shut, shuts it down. Another very popular way of shutting something down is to have sort of this tethered ball that swings in place. So here the channel is open and allows flux, and then here this ball swings into place and plugs up the mouth of that channel. And what that might look like is shown here. Here you have sort of a representation. So these are the transmembrane domains of an, let's say, a channel protein. It's sitting in the membrane with this, in this case, 4 alpha helical structures, and it's forming this pore. So it's forming this little mouth right there. And then there's usually a gating mechanism that is able to allow flux when it's open or plug it up when you don't want the flux. Now, before we move into carrier-mediated transport, let's just take a break, and I'm just going to start another recording that will start then with carrier-mediated transport.